All right. Thank you very much for attending the Research Horizon events. And my name is Haixu Tang. I'm professor of informatics computing here at our school. I'm doing basic bioinformatics research, and today I'm going to talk about our research on algorithm for computational mass spectrometry. So in the last 20 years, the most exciting progress in life science and biomedicine is application of the high throughput technology to study not a single molecular or even a few molecular, but all the molecular inside a cell as a whole. So for example, in 2000 and 2001, there's an announcement for the completion of Human Genome Project, which characterizes the entire genetic information or the whole set of gene or so-called genome in human cell. That basically gives us actually the very rapid progress of the new field called genomics. It's all because of this new technology advancement in DNA sequencing. There's even more advanced since then. Was the HGP, the Human Genome Project, finished within like $1 billion at least, and now you can sequence one human genome with less than $1,000. Okay, but biologists or biomedical researchers are not only interested in nucleic acids, they are interested in other kind of molecules like RNAs, like proteins, sugars, metabolites, and lipids, and many other things. So they want to also study the whole set of those molecules inside the cell. And so basically there are other fields like omics field, which we call proteomics, studying the whole proteins, glycomics, studying all sugars, and so on. And all these technology, all these kind of fields were made advancement depending on the same technology called mass spectrometry. Mass spectrometry is a chemical technique that can measure accurately, very accurately, the molecular weight of a large molecular, and based on this, you can basically get a signature of this different kind of molecular, and use the signature to identify, characterize those many different molecules inside the cell at a very high accuracy and the resolution. And also, furthermore, you can also quantify, get the abundance of these molecules inside the cell, and monitor them, their changes, iterations across different environmental and other conditions, for example, disease versus non-disease patients, and so on. So what my lab is focused on doing is for mass spectrometry, develop algorithms and statistical methods to help automated analysis of mass spectrometry data, particularly in the field of proteomics. So there are 4 different important problems can be addressed by mass spectrometry proteomics, namely to identify proteins in a complex mixture and to quantify those proteins, called protein quantification, and also to study modifications on proteins, such as the most complex modification called glycosylation, which adds a glycan molecule on certain amino acid residue of proteins. So we have in the past like 10 years or 15 years, my lab has developed many different algorithms and tools to address these different kind of problems. And more recently, we also try to develop methods for addressing the application of mass spectrometry for the influence of protein interaction, to study basic protein-protein interaction inside a very complex mixture. So many of this work also actually collaboration with my colleagues in bioinformatics group. So for example, as a proteomics work I presented in my last slide was a kind of collaboration work with Pedja Radovac, who is an expert in machine learning. So we developed a machine learning approach for that. And we're developing, also collaborating with Jank Sahinap, professor in our department, who will study algorithm for cancer genomics. So we developed proteogenomics, a method algorithm from mass spectrometry data and integrate them with DNA sequencing data. And we also study using mass spectrometry to study microbiomes, which is, you will hear more from the next talk, just following me, from Professor Yuzhen Ye. And so we do some metaproteomics work too. Okay, so in addition to the computational mass spectrometry, my lab also have a few other projects. So we do develop privacy-preserving technique for human genome data analysis and sharing. We also study multiple genetic elements and microbiome genomics. and some epigenomics in plant biology. And that will conclude my talk. And thank you very much for your time.